The first speaker is Steve Benner from the University of Florida at the Foundation for Molecular Evolution. Did I get that right, Steve? I came close anyway. Happy to have him talk and go for it.
I never correct the people who introduce me. Yeah, I'm going to try to first start by doing what's necessary to do for an audience that is not necessarily in the field that I'm going to talk about, which is to say what I'm going to say first. And what I'm going to say is the following propositions. First, sort of as the guiding theory proposition, that a polyelectrolyte with a uniform structure is a universal for Darwinism. And I haven't seen any of those cards go up, but maybe that's because they're not out. A polyelectrolyte means a molecule with many repeating backbone charges. So DNA, in your case, is a repeating minus, 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 minus backbone charge. In principle, on an alien planet, you could have a genetic molecule with a repeating positive charge in the backbone. But the argument is, the proposition is, and it's the guiding theory for this talk, is that a polyelectrolyte with that uniform structure will be universal in life and water, no matter where you find it. The second thing is a specific hypothesis to provide context, is that we assume in this talk that the polyelectrolyte that supported the first Darwinism on Earth was actually ribonucleic acid, RNA. We'll have a lot of talking about that. The next thing we're going to do is we're going to structure problem selection. And this is a very important question, right? This is a huge field, and people just wander through it saying that they're working on the origin of life, when actually what they're doing is having fun with something that they struck their fancy, and perhaps even if all the work goes exactly as planned, everything turns out well, they won't have actually learned anything about the origins of life. So what we do here is try to come up with things like the proposition that settled science that says that RNA is impossible to form prebiotically. And if your guiding theory is this polyelectrolyte theory, and your guiding hypothesis is that RNA was first, if settled science says that RNA is impossible to form prebiotic, you have a paradox. You got to address that. And that focuses your research on specific problems and leaves all the other stuff aside and not relevant. So then we go back to strategies for paradox resolution. You'll discover that in this field, settled science for the longest time has not considered the mineral environment of the processes that might allow to form RNA to form. And in fact, the way paradoxes in the real world really are resolved is they're not really paradoxes. You've just forgotten to say something in your line of assumptions or your line of premises and whatever syllogism you think is true. And mineral-guided processes actually are ways around this paradox, and we'll talk a little bit about that. The next thing I'm going to tell you about is a natural history context. It turns out that the needed chemistry-mineral combination was very short-lived, was very transient on Earth. And if you stick through this with me, your reward, and this is always important, right, just to let out the reward, your reward will be a relatively simple path to form RNA prebiotically, a relatively narrow date when life on Earth originated prebiotically, and a relatively clear statement about the next rounds of paradoxes that need to be solved to bring this forward. And so these are the people who I will cite frequently throughout the talk, as well as people who have been cited on publications, and we're especially indebted to people like Elisa Biondi and Hyojun Daniel Cooter in my laboratory, who have been doing a lot of the chemistry, but a lot of input from Steve Moises and Ramon Bresser and Dustin Trail, who's here, and Kevin Donnelly and David Catling have been to some extent our adversaries, or at least critics of the process. And Rob Levinsky is my mineral dealer, and so I have also bought a lot of rocks, but I probably won't pass them around because the audience is too large for that to be working well. All right, are you ready? This is going to be a long morning. Great. So let's talk about the guiding theory, that these polyelectrolytes with a uniform structure are universal for Darwinism. And for those of you who actually know the structures of DNA and actually were forced to memorize it, there's a piece of DNA, and the repeating linking groups are phosphates, and they carry a natural negative charge on them at neutral pH. And that's what we mean by a polyelectrolyte. It's a repeating structure with minus, 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 minus. Now, it's always quite surprising to people when they realize that in your DNA you form a double helix, where a backbone minus, 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 minus binds to another backbone minus, 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 minus. And the students always say at some point they realize, well, wait a minute, Coulomb's law says that pluses attract minuses. So what are we doing in trusting our genetic information to a polyanion binding to another polyanion? That seems to be a repulsive interaction, and indeed it is. But it actually turns out it is responsible for Watson-Crick pairing rules, because when these two strands get together, the repeating backbone negative charge in one strand repels the backbone from the other strand and forces the interaction between those two strands as far away from the backbone as possible. And that's where we get these rules that you learned in 8th grade, where A pairs with T and G pairs with C. You make an analog of this molecule, and we've done so, without the repeating negative charges, the molecule doesn't have those rules at all. And further, the backbone negative charges causes this molecule to not fold, right? Because by extending itself, you bring those backbone charges far away. Let me put another picture up, right? So it's this backbone repulsion that forces the base-base interactions as far away from the backbone as possible. And it also forces the strand to unfold, and that's needed for templating. That's how you get another molecule of DNA, or RNA for that matter, synthesized on a polyelectrolyte backbone. And so this means that for physical reasons, this repeating negative charge in the backbone, here represented by those P's with a little negative charge on them, is not quite as stupid as it might seem from first glance. But it's more important than that. Experiments support this, and I know I'm just going to throw up on the structure of this slide variants of this molecule that have been made in our laboratory and other people's laboratories without the repeating charges, and these things fail. Now, you could say, Stephen, if you just had been a little bit more clever and made one of the more interesting or correct molecules, it would have worked without the repeating backbone charge. But at some point, you know, you stop making these molecules in part because the funding agency doesn't tolerate it anymore. But this is an empirical basis for what I told you a moment ago, which is really just sort of charge Coulomb theory, and this is how we know that this general rule is likely to be universal. Now, there's another reason for this, and this is actually perhaps even the more important thing, is because Darwinism is equated to life, and life is equated to Darwinism at some fundamental level. Now, it's a theory, right? We believe that Darwinism, that is this process of reproduction with errors, where the errors themselves are reproducible, is the only way in the universe for allowing organic matter to self-organize, to give properties that we value in life. And that means that even if you get on the Enterprise and go to a distant planet, you know, you're going to find organisms that are organized in their chemistry, and they got there by this process of random variation, non-prospective random variation with errors where the errors themselves are replicable. And that's actually a difficult thing to get. And so I just put up here some well-known things in chemistry which may not be well-known to you, but really you have to be able to change the sequence of your of the bases of RNA, A, G, C, and U, actually is constrained now to a very short period of time, not even 1 million years, it's a 500,000 years. So this plus minus is a statement about the physics of the atmosphere returning to an unproductive state from a very transient time when the atmosphere was productive for forming the bases. In fact, the biggest source of uncertainty is the availability of dry land, which we discussed yesterday from June Kornaga. I have, by the way, taken this out of Kornaga's paper with Roses. I have reversed the time scale, so Kevin likes it from old time to modern time. And of course, there is this very nice plot which gives us, you know, a reasonable amount of the least crust. That is 1.0, not 0.1, because we flipped it. Remember, of course, the amount of crust is not the amount of dry land, and but on the other hand, reducing the oceans with impactor iron to hydrogen, dihydrogen, will certainly dry some land out. And the question is only what the temperature is of that dry land under whatever atmosphere is doing whatever to the greenhouse effect. So there you go. So what I've done today is basically told you the guiding theory. Polyelectrolytes are important. We've told you that we are hypothesizing that RNA was the polyelectrolyte that formed the first Darwinism on Earth. We focused on settled science paradoxes that claim that RNA could not have formed. We've given you mineral-based solutions for these things. And by the way, just for the record, I brought with me some prebiotic soup. So if you would like to know what was almost certainly on the surface of any dry land that June can give us, all right, there it is, and you can do whatever you want with it. We even have a little bit of the nickel and from the meteorite impact, which you can actually take back to the lab and make RNA out of. You need a natural history context. I've given you one scenario which is involving impact, very similar to what Kevin told you about. And now it's time to collect your reward, a relatively simple path to form RNA prebiotically, a relatively narrow date when life on Earth originated prebiotically, and a clear statement of the next round of paradoxes. And for that, I mean, I just mentioned a few of them. We don't know enough about this borate-controlled carbohydrate processing. We need to know more about that. We actually need to know more about the availability of Hadean borate, and I'm hoping that Dustin will tell me about that. We've not said a word about chirality here, and that's a big problem. And there's a range of paradoxes that also arise from the poverty of RNA as a catalyst. It's actually not a very good catalyst. And when it is a catalyst, it tends to destroy RNA more than it makes RNA, but that's a detail. So there's what you said, but let me just make a comment about, you know, multidisciplinarity. I greatly love this. What we've done here is we've given you one of my favorite slides. I've been using this now for years. Oops, I didn't want to do that. Let's go back. We've used information from natural history to talk about why we are looking for RNA. It has to do with the fact that the RNA ribosome machine is RNA. We've done a lot of synthetic biology to try to explain how this works and to confirm theories by doing synthesis. We've focused on this. We've used the synthesis to also help go out and find an alien life form. You know, in my career, what's been striking to me is how many times areas that were considered to be separate disciplines have merged into a single discipline. And it's not by getting conferences together where someone who's an expert in geology talks to somebody who's an expert in chemistry. It's by having students trained so that they move freely from biology to chemistry to geology and to various other things. And that is, of course, your challenge for the students in the audience. So let me stop there. Thank you for your attention. I'd be happy to answer any questions you might have.
We're open for questions.
Yeah.
Yeah, so going back to some of the topics that you had at the beginning of the talk, since you can now make alternative genetic languages in the lab, which is really, really exciting, have you learned from that process about why life might have chosen the specific nucleobases and the number of nucleobases?
Yeah, excellent question. And the answer to that is, yeah, so the question is whether we can now explain why you use G, A, C, and T and not some of these other ones that we've made. And you got to remember, people are easy to fool, and the easiest person to fool is yourself. So I can come up with an explanation for that, and we've got volumes. We've written lots and lots of papers based on the experimental work with alternative systems. But you got to remember, when you ask an organic chemist, you take an organic chemistry course, you walk up to the professor afterwards and say, hey, why is this way? And the professor will give you an answer. And then you say, oh, no, wait a minute, it's exactly the opposite. and the professor will give you just as convincing an answer if it's the opposite. And that's how you know that organic chemistry is a science. So it would be better. So we have a great paper from Urs Sassmari, who actually went out and said, yep, 4 is the optimum number of pairs, and he had an argument for it. It would have been much more comfortable for me if he did not know before he made that argument that the natural stuff had 4 bases. So the answer is yes, we've got lots and lots of explanations, but this is a just so problem. You have to worry about whether we can come up with something independent to justify our favorite theories. Yeah. Well, to get something that a chemistry flask, a material that has to get out of the flask, go out into the, down to Lake Avenue and buy itself some food, right? That's what you're talking about here. Or to photosynthesize, or to do any of these other things that get you the food, the resources. We in the laboratory are feeding this nice stuff. So you're all of a sudden talking about compartmentalization. We don't know how to do compartmentalization. You're having to talk about sensing, right? We had to move in the gradient of a food. Photosynthesis, where's Ken? Oh, there you go. I mean Blankenships. You're going to hear all sorts of, these are hard problems. And if I knew how to do photosynthesis with RNA catalysis, I would be, I don't know what I would be doing. I wouldn't be maybe standing here. So there are lots and lots of, like, going from just a system that can create copies of itself with errors, where the errors themselves are replicable, is a long way away from being self-sustaining. Yeah, I'm in the back. No? Yes? Sure, why not? Are there other people? Yeah, I know Phil Holliger is the better person here than I am. So we cheat. We take natural DNA polymerases, but guess what? The natural DNA polymerases have evolved for 4 letters, and they don't take 8-letter anything. So we have to then mutate, evolve. We do a lot like what Francis Arnold, who is here at Caltech, we do directed evolution on these DNA polymerases. Now, Phil is the guy who really is doing a good job of this. He is actually getting the RNA molecule to itself be the enzyme. So he does not have to go to Sigma, Aldrich, Promega, or any of these other companies and bring in a polymerase that doesn't want to work anyhow and have to mutate it. So clearing this cycle, getting this replicase going where the RNA molecule is itself catalyzing the replication, that is Phil Holliger, and he is somebody who you should have invited to this conference instead of me.